In this video, we'll start to look at cells, and we're going to start by with some basic definition of the shape of cells. Now, there's over 200 different types of cells in the human body, and many of them are quite different shapes. So not surprisingly, there's various names that are assigned for different shapes. And let's go through it and get some of the basic nomenclature of the shapes of cells. Squamous means thin, flat, and scaly. Cuboidal, many of these make sense. Cuboidal is squarish looking. Columnar look like a column. They're taller than they are wide. Polygonal cells are irregular, like a polygon. Stellate, star-like. Spheroid or ovid are round to oval. Discoid are dish-shaped. Fusiform. Fusiform means thick in the middle, tapered towards the ends, and it's kind of this shape. So thin at one end, thick in the middle, and thinner at the other end. And then we also have fibrous, which is like a thread. Now, here's just a visual representation of the different types. Squamous for the flat ones, cuboidal because they are cube-shaped. Columnar, they're taller than they are wide. Polygonal, they have the multiple sides to them. Stellate, very star-shaped. Spheroid. Discoid, great example are red blood cells. Fusiform, we'll see that with some of the muscle cells. And fibrous. Now, how big are they? Cells are limited as to how big they can be because of the surface area to volume ratio. You get to a point where you make a cell too big, and its surface area relative to the increase in the volume of the cell doesn't keep up. And of course, the cell membrane is about transporting things in and out of the cell. Give you an idea, most cells are about 5 to 15 micrometers in diameter. Now there are some that are bigger than that. The largest is a human egg cell at about 100 micrometers. Now some nerve cells, some neurons, can be about 1 meter in length. Now they're very thin, but they can be quite long, and these are the ones associated with the spinal cord. And we'll see them when we get to the nervous system. As I mentioned earlier, the limit of size cell, there is a limit, is that what happens is as I keep increasing the size of the cell, the volume increases more than the surface area. And so, because volume is proportional to the cube of the diameter. And as the cell gets bigger, more volume that you have to get wastes out of, nutrients into, the relative size of the cell membrane does not keep up. So cells can only be a certain size and still function. I will not go through the calculations. If you want to, feel free to prove it to yourself that indeed as the cell gets bigger, the volume increases faster than the actual surface area of the plasma membrane. Now, how do we know about cells? Of course, as we said, you can't really see them with the naked eye. And of course, light microscopy has been the standard way, but it has its limitations. You can see the plasma membrane, nucleus, the cytoplasm with a very high-end light microscope set up properly. Transmission electron microscopes, they have a higher resolution. They can see more details. We'll see some images from these. And scanning electron microscopes, they're about surface features. They look at surface features. And we'll see some images from all three of these as we go through the course. Just give you an idea, sort of the clarity. Here is a image from a light microscope. You can see that there certainly is a degree of fuzziness to it. And it has to do with the limitations of the optics and the use of light. Whereas here you can see a electron microscope. It's because of the physics of the electron beam, etc., and the magnets that are used for the electron beam. It gives us a higher resolution. And a resolution is the ability to clearly distinguish two objects as being separate and what the distance is between them. So, most of you are quite aware of the basic components, and we'll have a look at some of them. Of course, the plasma or cell membrane, and of course, this is a very important part. It's a plasma membrane that defines the boundaries of the cell. It also is the surface in which wastes leave and nutrients come in. It's also the way that the cell interacts with its environment, the material outside the cell, between cells, how it interacts with other cells, etc. Not surprised, we're going to see there's a lot of receptors on there that take or can listen for incoming messages. So it's a very active, dynamic surface. Don't think of it as sort of the outside of a chicken egg that's just solid as a boundary. This is a very dynamic place where much happens. So it surrounds the cell and defines the boundaries. It's made of proteins and lipids. And inside this, we have the cytoplasm and we have the organelles, cytoskeleton, inclusions, talk about those, and the cytosol. Now outside the cell, we have the extracellular fluid. And we'll talk about what it is. It depends on the tissue as to what's exactly in the extracellular fluid and how much there is. And when we get into histology, we'll talk about cells versus their surrounding matrix, which is the non-cellular component of a tissue. A very basic diagram of a cell. I want to take a second to point out, not all cells look like this. Cells come in all sorts of different shapes. We'll certainly see this once we get to the immune cells or into the nervous system cells. They have some very unique shapes. And just a reminder, not all cells have all the organelles we're going to look at. Something like a liver cell will need certain types of organelles more than other cells in order to do its work. Muscle cells tend to have a lot of mitochondria in them because of the demands of energy. So this is a very much an idealized cell where they're trying to show all the different organelles that could possibly be in there. As I said, many cells in the body don't have all these organelles, or they have them in different ratios. So let's just talk a bit about the plasma membrane. The plasma membrane is the border of the cell. In light microscopy or in electron microscopy, you see it as dark parallel lines. I should say electron microscopy. In light microscopy, you'll just see it as sort of a blurry sort of boundary. You have to go to electron microscopy and actually see the double parallel lines. And not surprisingly, that if we have a cell, there's a basic cell, and that was the membrane, that there's an inside, there's an inside surface to the membrane, and there's also an outside surface to the membrane. So there's the intracellular and extracellular faces. So part of it faces into the cell, and part of it faces into the outside world. So basically, what are the functions? It defines cell boundaries, governs the interactions with other cells, and it controls the passage of materials in and out. Here we can see an electron microscope image. You can see here is the cell membrane. You can see the double layer of it. And in a previous chapter, we talked about the phospholipids, how the phospholipids have a hydrophilic and a hydrophobic end to them. And we'll get more into that. Here you can also see the nuclear envelope, the nucleus, the chromatin, etc. Now, when we're looking at a plasma membrane, as I said, this is not a solid structure. It's a very dynamic, it's very fluid. Often we hear it called the fluid mosaic model. That what we have are these individual phospholipids. So these are all different individual phospholipids. If you don't remember what those are, go back to the previous chapter with the hydrophilic ends, because of course I have a lot of water out here, a lot of water in here, so the hydrophilic ends like to line up there, and the hydrophobic ends tuck themselves away from the water. So they really do self-assemble. Some of the other characteristics we can see here is we can see right here, these are cholesterol molecules. They help to stabilize the membrane. Otherwise, the membrane would be very liquidy. It is somewhat liquidy, but it'd be even more liquid-like. So cholesterol that's stuck into the cell membrane helps to stabilize it. Then we have a number of proteins, and we'll walk through them. in a bit, but you can see an example here. Here's a channel protein. This allows material to pass out of the cell or material to pass into the cell. Some of the proteins actually are on the inside of the cell membrane. They pass through the cell membrane to the outside. And if you're a protein that's both inside and you also stick outside and you go all the way through the cell membrane, you're a transmembrane. If you're a protein, which is just on one side, here we are, this is on the inner side, you don't go all the way through, you're a peripheral protein, and I can have peripheral proteins on the outside, they don't go all the way through. Also, it's not just about proteins, often we have these conjugated molecules. So in this case, I have a protein, and attached onto the protein is a carbohydrate. And so it is a glycoprotein. So this is the carbohydrate portion, and this is the protein portion. We also have cases where I have, here is a phospholipid, and attached onto it is a carbohydrate chain, and that's a glycolipid. So you can see that there's a lot of things. In many ways, the outside of a cell is very fuzzy. It's got a lot of things sticking out, glycolipids, glycoproteins. And then we have all the other different types of proteins. As I said, the channel proteins, the transmembrane proteins, the peripheral proteins that are either just on the inside or just on the outside of the plasma membrane. We also have, just to remind you, the lipid bilayer, and then we have the cholesterol that's embedded. So this is a very good overlook at the cross-section of the plasma membrane. Now the membrane itself is about 98% are lipid molecules. And not surprising, the vast majority of those are phospholipids. That's about 75% of the membrane lipids are phospholipids. And just to remind you, they're empathic. So they have both a hydrophilic and a hydrophobic. The hydrophilic phosphate heads face towards the water side, which is either the inside or outside of the membrane. And the hydrophobic tails orient themselves towards the inside. Now, they do have the ability to drift laterally, so the membrane is very fluid. We also have some other membrane lipids. We have cholesterol, which we showed you. That's maybe about 20% of the membrane lipids, and what this helps to do is it helps to stabilize the lipid bilayer. Now, so it doesn't become too liquidy, otherwise it would just fall apart potentially. So you just need enough cholesterol. You don't want too much, you don't want too little in the cell membranes. We also have the glycolipids. They're about 5% of the membrane lipids. The phospholipids, they're phospholipids with a short carbohydrate chain attached onto it. Also, a lot of this fuzzy stuff that we talk about sticking outside, it has a special name, and it is the glycocalyx. And the glycocalyx is the carbohydrate coating on the outside of the surface. It has a big role in basically identifying the type of cell, interacting with other cells, defining that this cell belongs to you for the immune system, and a whole number of other functions. Let's just quickly look at some of the membrane proteins. That when I look at proteins, they're only 2% of the molecules in the membrane, but proteins are big. They weigh a lot, so they actually make up 50% of the weight of the membrane. Now we have different types of proteins, which we had a look at in that diagram. There's the integral ones. These ones penetrate the membrane. And we saw some examples of that. We saw transmembrane, which pass all the way through. And if you're going to be a transmembrane protein, you have to pass through the phospholipid bilayer, where part of it is hydrophilic and part of it is hydrophobic. So the protein has to have hydrophilic regions that will correspond to the hydrophilic regions of the lipid bilayer, and hydrophobic regions that can pass through the center, which is the hydrophobic part of the lipid bilayer. And some of them can drift along in the membrane, but some of them are anchored to the cytoskeleton. And let's just back up. I just want to show you what the cytoskeleton is. The cytoskeleton is, the name actually kind of suggests what it is. You see inside that there's this little fibrous network. It looks very much like a skeletal network. This gives shape. It does a whole number of things for the cell, but one thing it can do, it can help to anchor the plasma proteins so they do not move. We also have the peripheral proteins. The peripheral proteins, they attach to one of the faces of the membrane. They don't go all the way through. If they're on the inside, they're usually attached to the cytoskeleton. Now here we can have a look at the transmembrane proteins. As I said, you know, here's the protein right here. Part of the protein here and here is hydrophilic, because it's hanging out with the hydrophilic part of the lipid bilayer, and this center part of the protein would be hydrophobic, which allows it to insert itself into this region, this region right here. Now, what are the functions? I'm just going to mention them briefly right now. We will come back in the next video and walk through in more detail. But there's a number of functions. We have receptors. So these are there to maybe accept hormone signals or other chemical signals. Some of them are part of the second messenger system, which we will talk about in upcoming video. Some of them are enzymes. Some of them are channels, some of them are carriers. Some of them act as cell identification markers to basically say to the body, what type of cell is this, and also let the immune system know what type of cell it is. And some of them are cell adhesion molecules, which basically help to hold the cell in its place in the group of cells surrounding it, or the cells and the matrix. Matrix is any of the stuff outside cells that exists in that tissue. So we're going to leave it there, and we're going to come back and we're going to pick it up in the next video and start going into greater detail as to what all these mean.